Transcription growth factor B and bone morphogenic proteins act on nanotranscription gene to activate stem cells differentiation. Stem cells can differentiate into cardiomyocytes, liver cells, cartilage, osteocyte, skin cells, spermatozoa, and brain cells. Tests for pluripotency is a test used to determine whether stem cells are capable of forming into three germ layers: endoderm, mesoderm, and ectoderm. Human embryonic stem cells brought controversy to the general public. It is against the ethical standards of the Church because it uses and kills the human embryo to produce stem cells. With that, they made an alternative way: the adult-derived stem cells and amniotic fluid stem cells. A brown male mouse was crossed to a brown female mouse and produced a blastocyst. Stem cells was taken from the blastocyst using a microneedle and was cultured in the lab. White male mouse was crossed to a white female mouse and produced a blastocyst. The the cultured stem cells from the brown mouse was injected to the blastocyst of the white mouse. Then it was taken and injected to a surrogate white mouse and produces a mixed-colored brown and white mouse. Thus, the mixed coloration indicates that the stem cells are capable and functional.
An area of research in stem cell biology focuses on how to produce pluripotent stem cells without destroying an embryo. This involves the nuclear reprogramming of somatic cells. The basic idea is to take an adult differentiated cell and reprogram it to turn it back into its early undifferentiated state. Currently, there are several approaches to nuclear reprogramming. Here we will discuss on three different approaches. First, we have the somatic cell nuclear transfer. Second is the fusion of somatic cells and stem cells. And the third is by the creation of induced pluripotent stem cells. Somatic cell nuclear transfer. This involves a somatic cell and an egg cell. First, the nucleus from the egg cell is removed, becoming enucleated, and the somatic cell nucleus is transplanted into this enucleated cell to form an embryo, from which embryonic stem cells are isolated. The second approach in nuclear reprogramming is the fusion of somatic cells and stem cells. In this scenario, a fibroblast cell is fused with a human embryonic stem cell to produce hybrid cells which display similar properties with human embryonic stem cells. The third technique is the creation of induced pluripotent stem cells. In mouse fibroblast cells, four genes were found to be able to generate embryonic stem cells. The four genes are OCT4, SOX2, CMYC, and KLF4, but these genes are not actively expressed and so they are induced through retroviral vectors. Once the four genes were expressed, the differentiated cell is reprogrammed into a pluripotent-like state. Other than the three approaches mentioned, a study from 2010 revealed that RNA molecules, which are transcripts for the same genes used in reprogramming, produce induced pluripotent stem cells